Hello students. Today we are discussing about development of the brain. First of all, we are discussing about the neurons. It has contained a cell body and axons, and the collection of the cell body in the CNS is called nucleus, and the collection of cell body in the PNS is called the ganglion, and the collection of axons in the CNS is called tract, and the collection of axons in the PNS is called neurons. Now, first of all, we are talking about glial cells. These are supporting cells of the neurons. These are five types: astrocytes, oligodendrocytes, Schwann cells, ependymal cells, microglial cells. Astrocytes, which help in the formation of blood-brain barrier and derived from the neuroectoderms. Oligodendrocytes help in the formation of myelin sheath and derived from neuroectoderms. Schwann cells help in the formation of myelin sheath and derived from neural crest cells. Ependymal cells, they line the ventricles and derived from neuroectoderms. And the microglial cells, they have the phagocytotic function and the mesodermal derivative. These are the structure of the glial cells. We can see that Schwann cells, oligodendrocyte cells, star-like astrocyte cells, which form the blood-brain barrier. And we can see that ependymal cells, this is from the ventricle lining. And we can see that microglial cells, which they have the function like phagocytotic. Now, moving to the next topic is the development of the neural tube, which form the brain and spinal cord. The neural tube formation occurs by the third week. During the third week, two important changes in the fetus, gastrulation and neurulation. Ectoderm help in the formation of neural tube. In the center, there is a notochord which gives signal to the ectoderm to form the neural plate initially. Now we can see the structure, a notochord structure, and the notochord which gives the signal to the ectoderm which form the a neural group structure, okay, and which further invaginate in the form of the neurofold. After some time, the neurofolds which form the neural tube, and the center is called neurocanal. Neural tube, neurocanal. Okay. Now, after the third week development, it forms the 25th day. If the defect in the upper part of the neural tube, it's closes by 25th day, is called the cranioneuropode, and the defect in the lower part of the neural tube, it's closes by 28th day, is called the caudal neuropode. Okay. Now we are discussing about development of brain. The caudal part, you can see that this is the caudal part and cranial part. The caudal part of the neural tube which form the spinal cord, and the cranial part of the neural tube which form the brain. Brain in the form of just like forebrain, midbrain, hindbrain. We can see the structures: the forebrain, midbrain, hindbrain. And forebrain form the telencephalon, diencephalon, midbrain form the mesencephalon, and hindbrain form the metencephalon and melencephalon, which further deviate, which form the brain. And the caudal part of the neural tube, which form the spinal cord. Thank you.